healthcare, and biotherapeutics. Biotherapeutics refers to the use of biological substances such as proteins, antibodies, and nucleic acids for therapeutic purposes. These cutting-edge treatments offer targeted and personalized approaches to combat diseases, harnessing the body's own mechanisms to deliver effective and precise therapies, revolutionizing the field of medicine and opening new frontiers in healthcare.
BioTherapeutics Research Lab at CSIR NCL. We majorly focus on designing the processes for various recombinant biotherapeutic proteins. So these proteins are used in the treatment of various immuno-oncological diseases such as various types of the cancers. And what we try to do is understand the complex nature of these therapeutic molecules. We generally help the industries to develop de-risk intellectual property-based processes for this category of various biotherapeutic proteins.
I'm Anu Raghunathan, a Senior Principal Scientist in the Chemical Engineering and Process Development Division. The Biotherapeutics and Healthcare Research Team at NCL facilitates cross-disciplinary research and develops state-of-the-art facilities for cutting-edge work on fundamental discoveries, industrial applications, and translational research. We are a vibrant intellectual community and brought in a completely new dimension in the COVID-19 pandemic here in Pune. Can we deal with the COVID-19 pandemic better than the Spanish flu of 1918 that killed 3 times the 18 million victims of World War I? As a systems biologist at NCL, I coordinated efforts to set up a genome surveillance lab to monitor COVID-19 variants like Delta and Omicron and report them to the government. We continue to monitor any prospective variant even today in Pune. We all took the vaccine shots, but does that really work against all the new variants of COVID? The Delta, the Omicron, and the new XBB? I coordinate a clinical trial that studies T-cell immune response and how long the impact of these vaccines that we all took, Covishield and Covaxin, last. And do they help against all the new variants that are coming up through immune surveillance?
Water-based epidemiology, also known as sewage surveillance, is a very useful tool to track microbial pathogens and disease-causing microorganisms well in advance. So, WB approach has been already used in the past successfully for tracking and monitoring of poliovirus, enterovirus, cholera, and other pathogens. So, when this situation came in 2020 regarding SARS-CoV-2, the wastewater-based epidemiology was very much essential because there was a lack of RT-PCR kits and genome surveillance approaches. One can extract RNA from the wastewater samples using CDC protocol, and the RNA can be converted into cDNA and set up by RT-qPCR, and the viral fragments can be detected. So what is the advantage of this technique is, although we know that SARS-CoV-2 mostly asymptomatic cases, and asymptomatic individuals serve as a source for spread of infection. So tracking of such asymptomatic individuals is very important in order to control spread of the disease in the community. As we know that whenever patient gets infected with SARS-CoV-2, the patient starts shedding these virus fragments in the wastewater from the day one itself. However, development of the disease actually takes 4 to 5 days depending upon the type of variant. In Delta, it took 4 to 5 days. In Omicron, it took very less time. And in the newly reported XBB lineages, it is taking very less time to spread in the community. So, wastewater-based epidemiology will be very useful tool in the future for tracking the pathogens in low resource settings where RT-PCR and genome surveillance or genome sequencing facilities are not available.
So this is our Oxford Nanopore technology sequencer. This sequencer is called as the Gridiron, and it's a very high throughput machine where we can actually sample 5 different libraries at the same time. Currently 2 libraries are being sampled, and the beauty of this technology is that while sequencing is going on, we can analyze the data real time. So what you are seeing in the monitor is all the ports in the instruments which are currently sequencing, We can also look at the quality of the sequences which are coming out of this machine in real time. And we can also see how much data we are accumulating, what is the size of the DNA that is getting sequenced, and how many sequence fragments the data is getting accumulated. This is a very versatile technology and very rapid, very cost-effective way of pathogen diagnostics like COVID or other pathogens.
Hello, I'm Dr. Ramrup Sarkar, Senior Principal Scientist of CSIR National Chemical Laboratory. Our group's main work is theoretical and computational biology. We are trying to understand the fundamental mechanisms of biochemical systems, the signaling and metabolic pathways, and also we are focusing to translate that idea into the healthcare and biotherapeutic aspects. We are mainly working on two major diseases, cancer and infectious disease like leishmaniasis. Our contributions towards healthcare is development of computational platform for understanding the disease mechanisms, forecasting the disease, as well as we are using the artificial intelligence and machine learning techniques to understand the cancer rate predictions, target identifications, and proposing potential treatment strategies. Thank you.
Hi, I'm Dr. Mahesh Kulkarni, Senior Principal Scientist in the Division of Biochemical Sensors, CSIR National Chemical Laboratory. Our lab works in the area of diabetes using mass spectrometric and proteomic approaches. In diabetes, despite development in diagnostics and therapeutics, many patients develop diabetic complications. In this context, our lab is working on protein glycation, especially to identify novel diagnostic markers such as glycated albumin, which can predict diabetic complications as well as prediabetes. The other aspect we are working is to develop glycation resistant insulin, which could act as a template for developing long acting insulin. And we also work on fundamental aspects of diabetes, especially our lab focuses on age rate signaling and its impact on developing insulin resistance. Thank you.